Okay, so here I'm going to talk about gene regulation in bacteria, specifically how operons work, which is a way for bacteria to regulate genes that are in a common pathway. So there'll be a cluster of genes that are under the control of a single promoter. Now, remember, there's going to be lots and lots of operons within a bacterial genome. So we usually don't draw it in the context of the chromosome. We just draw a little section of the chromosome showing the operon. So in this case, again, we have a single promoter and it has a special sequence called an operator. The operator sequence is where the regulation is really happening. Then there'll be a single start site and a single mRNA transcribed from this promoter. Now that mRNA is going to be translated into multiple protein products depending on the number of genes within the operon. So again, these operons are usually clusters of genes that are involved in the same metabolic pathway. So the examples we use are the lac operon, which has the genes required for lactose metabolism, and the trip operon, which have the genes that are required to make or synthesize the amino acid tryptophan. Okay, another important component of how operons are regulated are an additional regulatory protein. So these proteins are going to be encoded by genes somewhere else in the genome. They could be just upstream or they can be far away. So these regulatory proteins have their own promoters and they will be transcribed into an mRNA, which eventually will be translated into this regulatory protein. Okay, and then typically what we're going to see is that under certain condition, this regulatory protein is going to bind directly to the operator sequence within the DNA. And this is going to block transcription. And so now we would see no transcription, no transcription, so no RNA made and no protein made. So this gene is essentially off, and we could consider this regulator a repressor because it's repressing transcription. Now, when that regulator is actually doing the repression depends on what type of operon it is. And that's why we use these two different examples because they're regulated in different ways. For example, the lac operon is an example of an inducible operon. So inducible operons are normally off, and they're only turned on in the presence of an inducer. So again, if there's no lactose, there's no expression from the lac operon. It's turned off. But when lactose is in the cell, the operon is turned on. Now we know that there's a little bit of more regulation than this that has to do with glucose. I'll get to that later. Now we can contrast an inducible operon with a repressible operon. And the example of this is the trip operon. Now, what happens with these operons is that they are normally on. So they are, they're biosynthetic pathways that are making something that the cell needs. Then they only turn off once that product has accumulated to high enough levels. So again, to contrast the trip operon with the lac operon, when there are low levels or no tryptophan, the operon is going to be on. And once the product of the pathway, which is again the amino acid tryptophan, the operon is going to be off. So again, like we saw with the lac operon, there's actually some fine tuning when it comes to the regulation, and we can talk about that later. Okay, let's take a look at the lac operon. Let's say that the cell has no lactose. Okay, so there's no reason to make all of these genes and proteins that are required to break down lactose if lactose isn't around. So where do we start? First, there should be repressor protein that's made from the LAC-I gene. Maybe I'll just put a big R there. Now, in this state, this repressor can bind directly to the operator sequence. And there were a bunch of experiments that showed that it actually binds directly to the DNA in a specific sequence. This blocks the promoter, access to the promoter, and there is no transcription, no translation, and so the gene is essentially shut off. Okay, Now let's say that some lactose is able to enter the cell. So now we've got the presence of lactose. What's going to happen is this repressor protein is actually going to have a higher affinity for the inducer than it does for the operator sequence. So I'm going to remove the repressor from the operator sequence, and we'll go make that back to normal. And instead of the repressor binding to the DNA sequence, it's going to bind to the inducer, which is allolactose. Allolactose is just a form of lactose that accumulates in the cell when there's a lot of lactose present. Now, when lactose is present and allolactose can bind to the repressor is no longer going to bind to the operator. And sometimes we indicate that with a symbol like this. Now, if that's the case, then there should be expression of these genes. Now, we know that there's further regulation if glucose is present or not. So let's just pretend that glucose is gone, and now there's going to be transcription of this gene. And a single mRNA is going to encode for the LAC-Z protein, which is beta-galactosidase or beta-gal, the permease, which is made by the LAC-Y gene, and then the transacetylase, which is the LAC-Y or LAC-A gene, which you don't really need to worry about. Okay, now these operons were figured out through the analysis of different mutations. So I'm going to make a little table. So I reorganized this so we can just think about the mutations that were looked at and what you would expect to see with respect to transcription of this gene, whether it's off and on or if it's inducible. So the first mutation that was used was the LAC-I mutation. So this is no repressor is made. So when you've lost the ability to repress the transcription of this operator, it's going to be on all the time, on all the time. Now, it's not on because allolactose is present pulling the repressor off. It's not on because there's no repressor, functional repressor to actually repress transcription. So the nature of it being on is a little different than what we saw when it's, when it's induced in a normal state. The next mutation is a mutation in the operator sequence, where now when repressor is made, so we'll say that the repressor is normal now, it's not the LAC-I. Now this repressor is no longer able to bind to the operator because the operator sequence has changed. And so now that since you're not allowing the repressor to bind, the operon's going to be on regardless if lactose is present or not. We call these states constitutive expression. It just means that it's expressed all the time, regardless if you have lactose or present or not. The other repressor is a LAC-I super repressor. So this is a mutation in the LAC. Let's get this all normal. This is a mutation in the LAC-I gene that makes it unable to bind to inducer. So regardless if lactose is present or not, the allolactose cannot bind. So maybe I'll block that there. So allolactose, the inducer, is no longer able to bind to the repressor, and that means that the repressor is always going to be on the operator sequence. So again, there's no way for that allolactose to bind to the repressor and pull it off of the operator. So again, maybe I should have erased this earlier, but when you have the repressor bound, you're not going to have any expression of this operator. And it's almost like activating the repressor activity of the repressor. It's super repressing. So this means that it's always going to be off. Now I want to build in this extra form of regulation. And this is due to the fact that bacteria cells would prefer to use glucose as their primary energy source rather than breaking down lactose. So what we're going to see is that when the cell has glucose present, so I'm just going to do plus glucose, the operon is essentially going to be off. So we can see how that would work with the presence of, or in the absence of lactose, right? Because the repressor should be able to bind and you're not going to get transcription. But if lactose is present, the repressor should be bound to allolactose and will be unable to, it'll be blocked from binding the operator. But the reason why transcription doesn't happen is because there needs to be activation of the RNA polymerase in order for really robust transcription to occur. So if I draw in an RNA polymerase like a blob like this, so this is a big enzyme that's going to start at the promoter, it's not going to be very stable when it's like this. And so transcription is going to be pretty lousy. There's going to be minimal transcription to where we can call it off. And if you remembered, I called that basal transcription. Just a little bit's made, but it's not enough to really see robust expression of the genes that are needed to break down lactose. And that's because the cell would rather spend its energy breaking down glucose first. So now let's take a look at what happens when the glucose stores are depleted. So the cell has used up all of the glucose. If lactose is still absent, there's no reason for the cell to make the genes that are needed to break lactose down. So again, repressor should be able to bind to operator and transcription should be blocked and you don't get the lac genes expressed. Now, when lactose is present, we know that it should be on, right? But in order for it to be on and express robustly, the RNA polymerase needs to be activated. So first off, the reason, again, just to be totally thorough, the reason why the repressor is not able to bind to the operator anymore is because it's bound to the inducer. It's bound to allolactose. Now, the RNA polymerase is going to be recruited to the promoter. And as we saw before, it's not going to be very stable unless there is another protein complex that has bound to the promoter sequence that can activate the RNA polymerase. So I'm going to draw it as these like lines like this. So this is that CAP protein. And remember, CAP binds to cyclic AMP. And what it's doing is stabilizing the RNA polymerase. And when that happens, we get robust transcription. You get all of your RNA, and then you get all of your different protein products made in high abundance. So that's why when glucose is absent, lactose is present, you see really strong expression from the lac operon. This video is already long enough, so I'm not going to talk about how this happens, how you can get activated CAP in response to reduced glucose. You can look back at your notes for that. But now let's move on to the repressible operon and see how that works. Okay, let's take a look at what happens with the trip operon. Now remember, this is a repressible operon. So normally it's going to start on. And so there's going to be expression of this gene, a single mRNA, which makes 5 different protein products. 2, 3, 4, 5. Those are all required to make tryptophan. This will happen in the absence of tryptophan. So without tryptophan, the operon is going to be on. Now, when tryptophan, the amino acid tryptophan, builds up, then this operon should turn off because it doesn't need to make any more tryptophan, so it should turn off these genes that are required. So how does that happen? Well, the TRPR, the repressor, is going to be made, and it's going to be hanging out like this in the cell. Now, when it's in this state, it cannot bind to the operator. So again, in the absence of tryptophan or a high enough level of tryptophan, the repressor is not binding. So now we need to figure out a way to get the repressor to bind the operator when there's a lot of tryptophan around. So this is where the product of this, pathway is going to, come back. Let's say this is tryptophan. Let's say when tryptophan binds to the repressor, it changes the shape of the repressor. and now the repressor can bind to operator. So when the repressor has changed shape upon binding to the product or tryptophan, it can bind to the operator, and that's going to block transcription from this gene. And so you're not going to make the transcript, the mRNA, and you're not going to make the proteins, and the cell's going to stop making tryptophan because it has enough. Now, once those levels of tryptophan decrease, the operator is going to have to turn back on, right? So then the tryptophan levels are going to decrease. This is going to come off. The repressor is going to change shape when it's no longer bound to tryptophan, and now the repressor is going to be off of the operator, and transcription is going to occur again. So this is a repressible operon. It's normally on, and it has to be turned off when the product builds up. Now, something interesting was revealed with the regulation of the tryptophan operon when the tryptophan repressor mutant was analyzed. So you would expect that in the absence of the tryptophan repressor, so we're going to delete the repressor because you're not getting any repressor at all. So your expectation should be that the operon is always on, constitutively expressed, because there's no repressor to ever repress it. But when they did this experiment, they found differences between when there's no repressor, there were differences between the absence and presence of tryptophan. And this revealed the importance of this leader strand. And what's happening here is something called attenuation. So even though in the repressor mutant, the operon is on in the presence of tryptophan, it's like really on in the absence of tryptophan. So the cell can turn it up even more. And there's that silly Spinal Tap video where he turns his amplifier up to 11 because it's higher than 10. So attenuation works at the level of transcription. It's not initiation of transcription, it's actually at the level of termination. So this is what happens. Initially, the mRNA for this gene, for the operon, will be transcribed. And what initially comes out is just that leader portion of the gene, of the operon. And the leader portion is special because it can form secondary structure. So if you remember, secondary structure in mRNA or in RNAs is actually hairpins. So there's different ways that this sequence can form a hairpin depending on the presence of tryptophan. So the secondary structure is dependent on the presence of tryptophan. So let's see how that works. So let's say tryptophan is absent. Remember here I said when tryptophan is absent in the TRPR mutant, there's a lot of expression of this. There's a lot of transcription and translation of this. So let's see, how does this work? So if tryptophan is absent and this leader strand is being transcribed, so the leader strand kind of comes out, there are a series of tryptophan codons. So there are 2 tryptophan codons in a row here. And what will happen is a ribosome is going to hop onto this. So I'm going to actually erase the trip so that it doesn't confuse you. So a ribosome is going to hop onto this mRNA, and it's going to try to translate a little bit of the sequence here. Now, when it gets to the tryptophan codons, if tryptophan is absent, it's going to stall. That's going to lead to the formation of the 2-3 hairpin. So there's going to be this hairpin formed, and that allows for full-length transcript to be made. Full-length transcript is made, then you get the genes, the TRP operon genes are going to be made, the proteins are made, and tryptophan is going to be made, which is ultimately the goal, right? Because if tryptophan is absent, the cell needs more tryptophan. So it's not that important to me that you remember the names of these stem loops, but this is called the 2-3 stem loop. Sometimes people call it the anti-terminator because it's not terminating transcription. It's allowing for full-length mRNA to be made. Now let's say that the operon has been on and tryptophan levels are starting to increase. Now the cell needs to sense the presence of tryptophan, even though maybe the repressor is not totally repressing the operon. Maybe it's because there's just not enough trip present, or it's because of it's in that mutant. So what we see now again is the 5' end of the mRNA is going to have a ribosome entry site. Again, it's not the one that's encoding for that's going to make the proteins of the trip operon. It's just making this little leader. Now, if tryptophan is present, the ribosome is going to be able to zoop through that little spot and then it'll fall off shortly after. But if it can move through that area fast, it's not going to stall and it's going to form a different hairpin called the 3,4 hairpin. So this 3,4 hairpin causes termination of transcription. So sometimes people call this hairpin the terminator. Okay? And here we're reducing the mRNA that's made, and so we should be reducing the proteins of the operon and eventually not making tryptophan anymore because there is tryptophan present. So again, tryptophan is a repressible operon, which means that it is going to be turned on normally, and then it gets turned off when the product accumulates. Now, attenuation is another way that the transcription can be fine-tuned from this operon. This is a common form of gene regulation for amino acid biosynthesis. Okay, hopefully that was helpful. If you have any questions, let me know.